Hey guys, it's really good to see you all, have you all again in the video with the fluorescence microscopy. So without making any further delays, let's start with our microscopy series. Okay, now you can see here we again have the slide from the previous microscopy series. Here actually I want you all to pause the video, remember the working of fluorescence microscopy again in your mind, and then pause and then play the video and then see if you missed out on anything. Go ahead, do it. I hope you have done it. Now, see, mercury lamp is a type of a lamp which will give us variety of wavelengths. Might be short wavelengths, long wavelengths, but we only want short wavelengths. Why? Because they are of high energy. Now, these short wavelengths, when all the light rays when pass through excitation filter, only short wavelengths passes through it, and they hit our dichromatic mirror and get finally reflected and pass through our lenses, behaving as objective lenses because this is epifluorescence microscopy, and finally hit our fluorochrome coated specimen. These fluorochrome absorb short wavelength radiations coming from the objective lenses and emit long wavelength light. But it is not important that they'll only emit long wavelength light, they might emit some other light radiations as well. So these when light radiations along with long wavelength pass through dichromatic mirror, are they meet another barrier called barrier filter, which will ensure that only long wavelength light rays hit our ocular lenses, and so we get our final image. Now, I hope the working and the parts and the principle of the microscope is crystal clear in your mind. But before going to the advantages of the microscope, I want you to get familiarized with what kind of sounds we make when we are talking about fluorochromes, because I know you must have heard about methyl orange, a common stain in light microscopy. If you haven't heard, no problem. When we'll be talking about some stains in the staining series, we'll deal every kind of stain, but just look at it how fluorochrome stain sound like. Okay, so we have most common acidic orange fluorochrome which stains DNA and appears orange in color. We have DAPI called diamidino 2-phenyl indole, which again helps in staining DNA, fluorescence green in color. We have fluorescent isothiocyanate, these attached to DNA probes, these attached to antibodies, and then green color is observed. And then we have trimethyl rhodamine isothiocyanate again, but it gives red in color. Finally, getting back to our advantages of the microscope, first is helps in differentiating live and dead cells in the specimen. Now, how are we doing it? See, whenever we are treating our specimen, we'll treat it with some specific mixtures of fluorochromes. Those fluorochromes will get attached to live cells, and some fluorescents will get attached to dead cells. So when the specimen is observed in the microscope, those live cells will appear different in color and dead cells will appear different in color. Helps in diagnosis. Diagnosis is what? If the bacteria or some kind of virus or some kind of antigen is present in the blood sample. How are we doing it? See, we are taking the blood sample, assuming it has some kind of foreign body or antigen in it, and then we are taking antibody which are beforehand attached with fluorochrome molecules. Okay, so when these antibody which are attached to fluorochromes are mixed with our sample, and then they are allowed to all kind of chemical reactions to occur, and then our specimen is gently washed with a gentle stream of water, and then it is observed under the microscope. If the test is positive, that means the antibody is present. How are we saying it? See, when antibodies were attached with fluorochrome, we know that antibodies bind with antigen molecules. And if antibodies are binding with antigen molecules and they are present even after washing the specimen, what does it signify? It signifies that antibodies do not want to leave the specimen because they are already combined with the antigen, with something, and that something is antigen. If antigen is present and antibodies are not leaving the specimen, and we can still see fluorescence in our blood sample under the microscope, that simply means the antigen and the antibodies present in the in the test, and the it gives a confirmatory test. And of course, this is a fast process. So I hope it is clear. Ecological studies, see, there are photosynthetic pigments which show natural fluorescence. So when these are hit by specific wavelength, and pigments are then differentiated on the basis of their natural fluorescence, and so we just have some kind of ecological studies. So these all are done by ecologists, those who want to study, and but they also help, fluorescence microscopy also help them. And we finally have some bacterial pathogen identification like Pseudomonas, Mycobacterium tuberculosis, Treponema pallidum. These all are fluorochrome stain. And then finally, when we see them under the microscope, we get to see their better image, better morphology, better detailing, zooming in, even better images. So that's how we will be identifying the bacterial pathogens. Moving on to the last slide, we are having three kind of images. I want you to look at them, observe them, and see what all you can see. We have a black background, right? We have some finely, other than normal colors, kind of bacteria or the or the organism present on the slide. Now these all are fluorescent light. Okay, that's how fluorescent look like under the microscope. First image is talking about live and dead cells, that as we talked about in the previous slide, live cells are appearing green in color, dead cells are appearing red in color. The second image is talking about mycobacterium. Now mycobacterium, we can see that it's rod-like, zooming in will give us better image, the colony characters we can make out, and the better detailing with the help of fluorescence microscopy is helping us to get all the details, right? And finally, you can see antibodies binding to specific molecules. You can see that we are having a black background and nothing else, only the antibodies which were attached to the antigen were basically are appearing in the ocular lenses, because antibodies are only the molecules in the whole sample which are stained with fluorochrome and nothing else, not even the antigen. It is just that we are ensuring that antibodies, if present in the sample even after washing it, this simply means that antigen is present. Now, with that, I hope this fluorescence microscopy, which appears to be difficult, which seems difficult, is clear in your mind. And if there's any doubt in the fluorescence microscopy, I really hope it's there. No another doubt. I mean, like, I hope I actually put so much into the fluorescence microscopy, so I really hope I explained everything that was possible and you are having no doubts. But still, if you're having the doubts are always welcome in this channel. I'll be replying to all of them as I'll be getting some time. Also, if you're clear with the microscopy series, please show your some enthusiasm in some comment section, so that I get to know that my work paid off. And with this, thank you so much, guys, for hearing me out. Let's meet another time. Bye bye. Take care.